Hello, welcome to Biology for You. In this video, we are going to discuss DNA structure. In the series of videos, we have been discussing different concepts in molecular biology. So, as a part of it, here we are going to discuss this structure of the DNA. So, basically, in this video, we are going to have the basic idea over it. That means what exactly the DNA is made up of. That means what are the building blocks of the DNA and how do they form different bonds to obtain that specific structure of the DNA. So the chemical bonds we are going to discuss. So basically, the DNA is a double helical molecule. That means there are two strands which are coiled on each other, and if you linearize the molecule, so it is getting this ladder structure. So basically what it is made up of, this is obviously a polymer. That means it is made up of two strands which are combined, and each one strand is a polymer of nucleotides. So basically these nucleotides are the building blocks of the DNA. So there are n number of nucleotides which are combined together into a single strand, and two of such strands will combine together and to make this double stranded DNA. So that is what DNA is made up of, nucleotides. So basically these nucleotides are combined together by phosphodiester bonds. So phosphodiester bonds are responsible for stitching up of these nucleotides together. So that means between two adjacent nucleotides you will find a phosphodiester bond. Likewise, these two strands are stitched together by hydrogen bonds. So basically these two strands are combined by hydrogen bonds. These nucleotides, all nucleotides are combined by these phosphodiester bonds. So in order to understand all these chemical bonds and structures, first we need to understand what are the types of nucleotides and what exactly is the nucleotide is made up of. So let us start with the nucleotide. As we understood, nucleotide is the building block of the DNA. So this nucleotide is the building block of DNA. It is again made up of a nitrogen base and sugar and the phosphate moiety. So basically this phosphate moiety is in the form of phosphoric acid, and there are 4 types of nitrogen bases and 2 types of sugars, pentose sugars present in nucleic acids, and the types of nitrogen bases in the DNA are adenine, guanine, cytosine and thymine. These 4 types of nitrogen bases can be found in DNA. And if you talk about the RNA, instead of thymine we will find the uracil nitrogen base. So what are the 2 kinds of sugars present in nucleic acids? Here we are going to have ribose and deoxyribose. So basically this deoxy form of ribose only present in the DNA, while ribose can be observed in RNA, and together these 3 together make the nucleotide. So let us understand. First let's go for the structure of nitrogen bases, followed by what are the 2 types of sugars, so that we are going to understand how all these are tied. So as we discussed, there are 4 4 types of nitrogen base in the DNA, and the other type of nitrogen base is the uracil. So let us start with the adenine. Adenine is basically a type of purine. So we'll find this purine ring. Purine is basically a heterocyclic ring. There are 2 cycles, and as it is a heterocyclic ring, along with the carbons we also find nitrogens in it. So basically the numbering will start from here: 1, 2, 3, 4, 5, 6, 7, 8 and 9, like this. So basically this is the basic structure of purine rings. So the numbering will start from here. I'm going to arrange these numbers so that I can draw hydrogen bonds in it. Okay, so adenine is basically a purine, that is 6 amino purine. So that is what the adenine is. This is 6 amino purine. So what about the guanine? Guanine is also a purine. So it is having the similar structure. You will find 2 nitrogens at 7 and 9th, as well as 1 and 3. This is basically 2 amino and 6 oxy purine. So that is what the guanine is. Guanine is 2 amino 6 oxy purine. So what about cytosine, thymine and uracil? These 3 are basically pyrimidines. So these are not bicyclic, but only one single ring will be present. These are also heterocyclic rings, and basically we'll start with uracil. So for our convenience we'll start with uracil. Uracil is, let us discuss the numbers. So basically for pyrimidine rings, the numbering will start from here, downward nitrogen. So from here 1, 2, 3, 4, 5 and 6. Likewise the numbering would be given. So here basically uracil is the 2,4 dioxy pyrimidine. So that is what the uracil is. 2,4 dioxy pyrimidine. And what is the difference between thymine and uracil? So that thymine can only be found in the DNA, uracil will be in the RNA. So the difference is both are similar, but along with these 2 oxygens, it also having the methyl group on its 5th position. So it is basically this thymine is basically 2,4 dioxy 5-methyl pyrimidine. So this is adenine, and 2 amino 6 oxypurine is the guanine, and 2,4 dioxypyrimidine is the uracil, and here is the thymine. Only we left with cytosine. So cytosine is basically also a pyrimidine. So here the 2 oxy position is common for all pyrimidines. present in nucleic acids, but the difference is here you will get a amino group. So that is what the cytosine is. So it is 2-oxy 4-amino pyrimidine. So these are the basic structures of nitrogen bases. So what are the sugars and phosphoric acids? So coming to that, as we already discussed, there are two categories of sugars, pentose sugars basically these are. So the basic structure of the sugar is like this. So basically the pentose sugar, that is ribose structure, is like this. So the numbering will start from here: 1, 2, 3, 4 and 5. So here for the sugars we call them as primes: 1 prime, 2 prime, 3 prime, 4 prime and 5 prime positions. So what is the difference between ribose and deoxyribose? So coming to that point, as name indicates, it is deoxyribose. That means one oxygen less than the ribose. So at 2 prime positions, if you don't find the hydroxyl group, that oxygen is lost, and if it is having a hydrogen group instead, then it is a deoxyribose sugar, and deoxyribose sugars are only present in the DNA. So these are the structures. So let us discuss how a nucleotide will form. So these are the components present in nucleotides: nitrogen bases and sugar. So let us talk about the nucleotides. So basically where the phosphate will attach, where the nitrogen base will attach to the ribose sugar. So that is going to be discussed. So here basically the 1 prime position, at 1 prime position the base, nitrogen base, will be attached on this ribose sugar, while the 5 prime position the phosphate moiety will be attached, and together it makes the nucleotide: nitrogen base, sugar and the phosphate moiety. So here how they attach. So basically we also classify nucleotide based on the number of phosphates. That means if one phosphate moiety is present, then it is nucleotide monophosphate. If 2 of such phosphates are present, then it is nucleotide diphosphate, and if 3 of such phosphate moieties are there in the nucleotide, that is nucleotide triphosphate. So likewise, based on the number of phosphates, we are going to classify nucleotides, and followed by based on the nitrogen bases, type of nitrogen bases. So if adenine is present, then it is adenine. So let us talk about the nucleotide triphosphates first. So if adenine is present, it is adenine triphosphate. If instead of adenine guanine is present, it is guanine triphosphate. Cytosine is present, CTP and UTP, and followed by TTP. So these are the types of nucleotides, and here this indicates the number of phosphates. So how do they combine together? So let us start with the phosphate, bond between phosphate and the sugar. So as we already know, In the pentose sugar, there will be a OH group, and this is the phosphoric acid. So basically phosphoric acid is this, and here is the pentose sugar. So here between these hydroxyl groups the bond will be formed. That means here one molecule of water will be lost, so that like this the bond will be formed between sugar and the phosphate group. So if another phosphate is there, that may be attaching to this first phosphate like this, so on. So here how a nitrogen base will be attached with this sugar moiety. So here let us start with adenine. So basically purines at their 9th positions. So here they will be attached to the 1' position of this sugar. So let us draw the structure of adenine. So here is the NH group, and here is the OH group. So again there is a loss of water molecule, so that the bond will be formed. So this is basically 1', 9 and glycosidic bond. So this is what the structure of nucleotide. So here this is the sugar moiety and this is the adenine. So it is adenosine monophosphate. So instead of adenosine, let us draw a guanosine. So both are the purines, so the structure is more or less similar. So this is what the guanine monophosphate. Here is the 1', 9 and glycosidic bond. Likewise, how pyrimidines will bind? Basically in the same way, the pyrimidines to their first position they will bind. So I'm drawing a structure of uracil. So here is an NH group and here is the OH and H. So the water molecule will be lost between them to form a glycosidic bond between deoxyribose sugar and this uridine. So this uracil forms the uracil monophosphate. So that is how the pyrimidines will combine with the sugars. So after this, let us also draw structure of thymine. So that is how TMP will be formed. So this is the structure of nucleotide. So let us talk about the bonds present between nucleotides, that is phosphodiester bonds, as we already discussed, between two adjacent nucleotides on the DNA strand. We are going to have these phosphodiester bonds. So how phosphodiester bonds will be formed? So let us start drawing a first nucleotide, and so we'll find nitrogen base over here. So let us draw structure of adenine. So this is what the adenine monophosphate is. So basically here is the H because it is a deoxyribose, and here is the OH at 3' position. So here for making phosphodiester bonds, this 3' OH group and 5' CH2OH or phosphate moiety are active. So basically the adjacent nucleotide, which is also having a sugar and nitrogen base. So let us draw structure of thymine over here. Okay, and also it is having a phosphate moiety at its 5' position. So between two phosphate moieties which are facing each other, the phosphodiester bond will be formed. That means there is a loss of sugar water molecule to make this phosphodiester bond. So that is how the phosphodiester bond will be formed between two adjacent nucleotides. So here is the adenine nucleotide, and here is the thymine nucleotide. So that is how the phosphodiester bond is formed. And coming to the other type of bond, that is hydrogen bond. How hydrogen bonds will be formed? So between two adjacent or opposite strands, between nitrogen bases, the hydrogen bonds will be formed, because this is the backbone. This phosphate and sugar moiety is the backbone for the DNA. That means they face external environment, but the nitrogen bases generally will be inside to the DNA helical structure. So what will happen? These nitrogen bases will find or face each other from the opposite strands, and they make hydrogen bonds. So here the hydrogen bonds are of two types, between adenine and thymine, and between guanosine and cytosine. So basically between adenine and thymine there are two hydrogen bonds, double bond will be formed, and between guanine and cytosine three hydrogen bonds, that is a triple bond will be formed. So how these triple and double bonds will be formed? So if it is an adenine structure, if it is an adenine structure, I'm drawing it in a opposite direction so that they will face each other, so the hydrogen bonds are easy to understand. So here is a NH2 group, and you will find double bonds over here, 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 and here, and this is 6-amino purine. So it should be in hydrogen bonding with thymine, that is present in opposite strand. So if it is the thymine, then what will happen? There is an oxygen and here is the amino group. So here one hydrogen present in this amino group acts as an acceptor, and here is the oxygen, so the hydrogen bond will be formed, and here between two nitrogen the hydrogen bond will be formed. So likewise two hydrogen bonds for each AT base pair. So A from the one strand, T from the other strand will face each other, and the hydrogen bond donors and acceptors will share their hydrogen bonds, and the double bond will be formed between them. So likewise how G and C bond will be formed. So let us talk about G and C bond here. So let us draw a structure of guanine. This is also in the opposite direction I'm drawing. So here is one nitrogen, so this is what the guanine is. So guanine will combine with cytosine in the opposite strand. So here the structure of cytosine is 4-amino 2-oxy pyrimidine. So here it is having, so this is what the cytosine is, and this is the guanine. So here is an oxygen, it will be involved in the hydrogen bond with one of the hydrogen of the amino group. One hydrogen from amino group of cytosine will be involved in the hydrogen bonding. Likewise between these NH groups the hydrogen bond will be formed, and between this oxygen and one of its hydrogen the hydrogen bond will be formed. Likewise 3 reactive groups and 3 hydrogen bonds will be formed between G and C, and it is A and T. So these are the hydrogen bonds which stitch two opposite strands together. So this is what this is how the hydrogen bonds will be formed, and that makes the double stranded DNA stable. And we also discussed the phosphodiester bonds which stitch two adjacent nucleotides together. So that is how the nucleotides are present and they combine in the DNA and followed by two strands of the DNA combined together. And here let us also draw a structure. Okay. So our convenience, it is like this. Between A and T, we find two nitrogen bonds. And likewise, we will also find phosphodiester bond between and this is the adjacent nucleotide. This is the phosphodiester bond and this is the guanine. So on the opposite strand you will find always cytosine. So again you will find a phosphodiester bond between two adjacent nucleotides present in the opposite strands. So here is a 3' group which is having OH moiety, and here you will find CH2O. So this is the phosphodiester bond between these two nucleotides, and this is the phosphodiester bond between opposite strand between two adjacent nucleotides on the opposite strand, and these are the hydrogen bonds. So this is how the DNA molecule is made up of. There are nucleotides, there are ribosugars, and there are phosphoric acid molecule. So likewise the DNA structure is made up of mainly two bonds, covalent bond, that is the phosphodiester bond and the other one hydrogen bond. Even though these hydrogen bonds are weak, together they give the strength to the DNA. Many of such hydrogen bonds between complementary bases, that are between A and T and between G and C. So between these complementary nucleotides you will find these hydrogen bonds, and even though they are weak, together they give strength to the DNA to be combined as a double-stranded molecule. So this is what the DNA structure, and hope you understand. If you like and share my video, and if you don't subscribe to my channel and press bell icon for the further notifications. Thank you.